So we are interested to compare DNA sequences from different species, different plant species, and we are looking at species that are, in a sense, very different. We look at a big tree called poplar, we look at a little flower called Arabidopsis, and we compare with species like grape or papaya fruits. So they are looking very different plants indeed, but we're looking at the level of the DNA where you can actually see a lot of similarities. And we know already that genes are conserved between different species, and you would expect those sequences to be the same. But we find that also sequences that are outside of genes are quite conserved. They have been kept for millions and millions of years, and we are working on finding those sequences and trying to work out what do these sequences do.
What we've done is we've written a computer program which finds such conserved regions of DNA between different species, and then we've tried to identify what is the purpose of why have these regions been conserved, what is the function of these regions. And in order to do this, we had to implement a variety of statistical methods, computational methods, to be able to do this on the scale of an entire genome. One of the first bits of software I wrote for this project was the core algorithm to compare two sequences and find what's similar between them. Continued to work on integrating this algorithm and other methods into a bigger software package, which is named Apples. And using the software package, we're now able to do this type of analysis on, well, any choice of species. We can run an analysis for an entire genome within 1 or 2 weeks.
And we found that there are actually hundreds or thousands of regions in the promoters of genes that are conserved. And these have been conserved over like 100 million years of time. So we think that the conserved non-coding sequences that we find are important in controlling the expression of the genes that they are upstream of. So they'll switch the gene on or off in the right tissue at the right time and under the right conditions. And we find that these conserved blocks tend to coincide with where you would find a nucleosome. And the nucleosomes themselves are thought to help regulate genes by controlling the accessibility of the DNA to transcription factor proteins. And so in that way, they're also thought to be regulatory sequences that we've found. One of the other results from the paper was that the genes that have the conserved regions associated with them themselves tend to be either involved in developmental processes in the plant. So this indicates that particular developmental pathways are perhaps more strongly conserved within the plant kingdom in general, and perhaps they're more fundamental processes that make a plant a plant. And also we find that another subset of genes which tend to have these conserved non-coding sequences in the promoters are themselves transcription factors. So it's the transcription factors that are regulating other transcription factors, which forms this idea of there being a core common network that's shared across different plant species.
The other positive outcome is that because these regions are conserved across so many different plants, means that the regulation is likely to perhaps to be similar, which gives us more hope that what we find out in Arabidopsis, which is a model plant that grows very quickly and just will grow in a small area, it gives us hope that what we find out in Arabidopsis we could actually relate to crops. The real value of this research for people who are interested in gene expression and understanding how gene expression is controlled is that it narrows down the regions of the DNA that you need to work on. So by identifying the regions that are conserved, we make the assumption that those are the important bits. And these regions are around 100 base pairs in length, or 100 to 200 base pairs in length. The other point that comes out from this research is the value of having multiple genomes sequenced. That some people might ask, well, why do you need to sequence other plant genomes? But by sequencing plant genomes, particularly ones at crucial sort of evolutionary distances, you can actually learn a lot more information. It's not just another plant genome.
So we want to study these sequences in more detail, and we want to work out what the regulatory codes are that make certain genes come up under certain circumstances, in certain tissues, at certain times. And we want to link the sequence pattern that we find conserved between the different species to the expression patterns that genes show. And we're making use of statistics, sequence analysis, and we also make use of visualization techniques in order to get a view over all the data set and get a visual perception of all the possible signals that we are facing in those sequences.